In case study number 3, we were asked to differentiate between innate and acquired immunity, differentiate between humoral and cell-mediated immunity, discuss each of the antibodies, including major functions and how they are produced in the body, discuss the differences between the primary and secondary immune responses, and to describe the different types of T cells and their function. Antibodies are made in response to an antigen and can recognize and bind to that antigen. They are produced by plasma cells. B cells are relatively soluble globulin proteins, and they are given the term immunoglobulins. The typical antibody monomer structure has 4 protein chains, 2 identical light chains and 2 identical heavy chains. The chains are joined by non-covalent bonds and covalent disulfide links to form a Y-shaped molecule. The stem of the Y molecule is also called the Fc, or crystalline fragment. So if you were to imagine the letter Y, that is what an antibody looks like. Each antibody has at least 2 identical binding sites for epitopes. Epitopes are antigen binding sites, and they are found at the ends of the Y's arms, and they're called variable regions. So if you were to look at a Y and you were to break it up into like a V and a stem, at the very tip of each end of the V would be the variable regions, or the antigen binding sites. These regions are identical to each other and identify with a specific antigen, and each antigen binding site is called a valence. For example, the human antibody usually has 2 binding sites, making them bivalent. There are 5 classes of immunoglobulins: IgG, IgM, IgA, IgD, and IgE, each of which is characterized by differences in structure and function. The first and most abundant antibody is IgG. IgG is a monomer and accounts for 80 to 85% of all the antibodies found in blood serum. IgG is located in the lymph and intestines and found in the surface of B cells. IgG is also secreted. There are 4 distinct subclasses of IgG: IgG1, IgG2, IgG3, and IgG4. Antigen memory response is based in the IgG class, where memory persists for a long time. IgG readily crosses the walls of blood vessels and enters tissue fluids to assist in destruction of pathogens. IgG can cross the placenta, conferring passive systematic immunity to fetus. The maternal IgG is the major antibody class found in the blood of a fetus or a newborn. The function of IgG is to protect against circulating bacteria and viruses, to neutralize bacteria toxins, to trigger the complement system, and when bound to antigens, enhance the effectiveness of phagocytotic cells. The second class of immunoglobulins is IgM. IgMs have a pentamer structure, which means there are 5 monomers held together by a polypeptide called a J, or a joining chain. IgMs make up about 5 to 10% of the antibodies in serum and are located in the lymph and on the surface of B cells in a monomer form. The IgM antibodies are the first antibodies produced upon B cell activation. IgM is relatively short-lived and appears as the first response to primary infection. This makes it valuable in detecting pathogens in a patient. It is likely that the disease is observed as caused by that pathogen. IgMs are secreted by plasma cells and generally remain in the blood vessels because it is too large to cross into the tissue. IgM is much more effective than IgG in causing the clumping of cells and viruses and in the reactions involving activation of complement. IgA has 2 subclasses. IgA1 is found predominantly in the serum and is usually in the form of a monomer, and IgA2 is found in body secretions as a dimer and is called secretory IgA. IgA is produced by plasma cells in the mucous membrane, and each dimer passes through a mucosal cell where it acquires a polypeptide chain called a secretory component that protects it from enzymatic degradation. The main function of secretory IgA is to prevent the attachment of microbial pathogens to mucosal surfaces. Secretory IgA is important for resistance in intestinal and respiratory pathogens, and IgA's presence in mother's milk, especially the colostrum, probably helps protect infants from gastrointestinal infections. These antibodies provide only passive immunity, however, and not systematic immunity, as they cannot cross into the bloodstream after the first 24 hours of life. The fourth immunoglobulin is IgD. IgD is also a monomer, and it makes up only about 0.2% of the total serum antibody. It's found in the blood, lymph, and resembles the IgG molecules, but has no well-defined function. So basically, they don't know exactly what IgD does, but they do know that it's involved in B cell activation and located only on the surface of early B cells. It never exists in secreted form or free form, and it's not a soluble antibody. The last immunoglobulin is IgE. IgE is a monomer and is secreted by plasma cells and bound to mast and basophil cells throughout the body. It's also found in the blood. These antibodies are slightly larger than IgG molecules, and they constitute only about 0.002% of the total serum antibody. Now, when we think about IgE, we think of allergic reactions. We also think about parasitic infection because they attract eosinophils to the site of parasitic infection. So, the IgE molecules bind tightly to their Fc stem regions to receptors on mast cells and basophils. These are the specialized cells that participate in allergic reactions. And also IgEs, their response can be protective as well, for it attracts complement and phagocytic cells, cytotic cells. It is generated in response to parasitic antigens, specifically designed to initiate an inflammatory reaction that preferentially attracts eosinophils to the site of parasitic infection, helping to break down the invading organism.